So, hello, everybody. I'm Stephen Gray. I'm a professor at UT Southwestern Medical Center in Dallas, and I'm going to try to expand on a talk that I gave last year on the basics of gene therapy, and this year just dig into a few more details. And I do have some disclosures. I do a lot of AAV gene therapy product development, and some of those end up working with companies and other organizations, and I have these conflicts of interest that have resulted from that. So what I hope to cover today is kind of a general overview, talking about a question that I get a lot when patients are trying to make decisions about how to move forward and what kind of treatments to go after and things. And one of this is looking at gene delivery as opposed to kind of mutation-specific personalized gene therapy approaches. So both of these, we have technology that can be applied, but kind of the pros and cons and differences of those general types of approaches. And then I'm going to focus really more on using adeno-associated virus, or AAV, as a gene delivery tool, and go into some aspects of that around why the vector DNA design is important and how we express the genes. A small overview on routes of delivery and safety issues associated with some of these vectors and some of these routes of delivery. And then what I want to kind of end up at the end to cover just quickly is how do you approach the evolving technology landscape? So do you go with the tried and true that we have right now, or do you kind of bank on newer technology that looks really exciting but may or may not be ready for prime time? And I just want to point out that there is some great past content available that ASGCT has put out. If you go to ASGCT's website, there is an area there specific for patient information. There's a Gene Therapy 101. There's also, I think in this meeting, there's archived sessions from last year. And if you have any trouble accessing those, you can email just meetings@asgct.org. Okay, so I'm going to give an overview of not all the types of gene therapy, but kind of the ones that might be relevant here to set some examples. We can have gene addition, and this is just where we're not doing anything to the mutated gene that's in the patient's DNA. We're just adding a gene on top of that. You can have RNA therapy. You can have ex vivo correction of stem cells. This is taking cells out of a patient's body, correcting them, and then infusing them back in to treat like immune disorders or blood disorders, as an example. And then antisense oligonucleotides and gene editing. And I've kind of separated these out a little bit because the top ones tend to be aimed at treating a specific disease. So a disease that is all caused by different changes in one gene. But these are not necessarily geared or tailored to specific mutations within a gene. And the latter two approaches tend to be aimed at treating specific mutations. So rather than trying to encompass all the different problems that can happen with a gene, they're really focused on treating a specific allele or specific mutation with a gene. And there's exceptions to all of these rules and everything, but just generally speaking, these are the types of approaches that are geared towards treating like an entire disease versus individual patient mutations. And I'm going to just back up a second and kind of give a basic background here that I'm going to build upon, and just remind everybody that we have DNA, and these are the genes. This is the instruction manual. This is like the permanent DNA that is in all the cells in your body. And then each of your cells use that DNA to make RNA. And this is a sort of temporary, transient kind of messenger, and the RNA carries the information that the cells read to make protein. And then the protein is what's doing everything in cells. Okay, and I like to give this example when we're talking about, just to illustrate different types of mutations, because this will be important to what types of treatments are geared towards certain types of mutations. And I've just gave a very simplistic overview of like what a gene looks like, where you have a signal that the cell can read, to say this is the start of a gene, and then a signal at the end, that the cell knows is the end of a gene. And then everything in between is broken up into these little 3-letter words. And so, all the 3 billion letters in your DNA are basically organized like this, where you've got your genes, which are the sentences, broken up into 3-letter words called codons. And so, just to make this a little bit easier to read, I've just transferred this into a sentence made up of 3-letter words that we can read, and it makes sense. It has meaning. The cat ate the rat and got fat. The end. Okay, so it has a beginning, it has an end, and the sentence makes sense, and it conveys a message. And so this would be like the normal or wild type gene. And then we could have 1-letter change, and that's called a missense mutation. And in this case, the cat ate the bat and got fat at the end. Instead of eating the rat, it is eating the bat. So the sentence still makes sense, but it has a slightly different meaning. So this might be called like a variant of unknown significance. There's a change, but we don't know if this is like causing disease or not. And then you can have the R in rat change to an E. Then the cat ate the eat and got fat at the end. And so now this is a different type of missense mutation, but in this case, it's likely pathogenic. Like the sentence doesn't quite make sense anymore. And then you can have other things called a premature stop, where you have a mutation, and here it's changing an A to an E, and that creates a signal that is the end of the sentence. And so now, the sentence is missing the end content, the gene is truncated, and so this would be called like a premature stop. And this could be pathogenic, right? And then you can have something where one letter is deleted. And if one letter is deleted, then the cell continues to read this sentence in 3-letter blocks. And so it shifts all the words in the sentence. And so now, after the cat, the rest of the sentence doesn't make sense anymore. So this is called a deletion, resulting in a frame shift, and this would be a severe mutation. You can have the same thing where you have one letter inserted instead of deleted, but it results in the same thing. Everything gets shifted out of frame, and this can be a severe mutation. And then the last type of thing that I was going to cover is a different types of missense mutation. And in this case, the cat ate the rat and got fit, the end. So instead of getting fat, it gets fit. And so this is a missense mutation. But in this case, the sentence still makes sense, but the sentence has a different meaning. And so this might be categorized as something where, you've actually changed the function of the protein that's coded here. And this could be like a dominant sort of toxic gain of function mutation. Okay, and then just to expand on this a little bit more, there's something called RNA splicing, where the message, the RNA is actually processed before you are able to make the protein. And so here I have a simplified sentence: the cat ate the rat, the end. Except when you actually look at the DNA, the gene, can be broken up when it's on the chromosome, and you can have these things called introns, or an intronic region that is just inserted in the middle of the gene. And so the different coding parts are broken up, and this is natural. Most genes are organized like this, and what happens is whenever you transcribe that DNA into RNA, then it'll contain that intron or the intronic region, but then there's signals that the cell will recognize that that intron should be removed. And so this RNA is processed by the cell where it physically cuts out that intron and removes it and then splices back together the two ends so that the RNA, becomes correctly spliced and then is read to make the correct protein. So you can have mutations that aren't actually in the coding area of the gene, they're in these introns, but it makes it so that these introns aren't removed properly or things like that. Okay, so the reason I went over this is because, talking about gene addition versus like sort of individualized treatments targeting specific mutations, this whole category of different types of mutations, all of these essentially result in a gene deficiency. None of these mutations are able to make the fully functional protein. So even though they're different individual types of mutations and they're in different locations, they all result in basically the same thing, that you can't make as much of this protein as the body should. And so all of those could be treated with a gene addition. So we'll contrast this with something like gene editing, where it's going to be very targeted to specific mutations, like correcting one letter at a time or a small group of letters at a time. And so now we've changed, and instead of having one treatment for all of these different things, now we have to make a separate design of a treatment for each of these individual mutations. And so just an example of this is, kind of an old report from a gene called MECP2 that causes a disease called Rett syndrome, marking different patient mutations that have been identified throughout this gene. So it could be attractive to do a gene replacement strategy where you deliver a functional copy of this gene, and then you have one drug that can treat all these different mutations. Or you could design 37 individual gene editing drugs to treat each individual pathogenic variant. And in the case of Rett syndrome, I think the challenge is if you do a gene replacement, you have to try to regulate this gene the way it would be naturally regulated, versus if you did a gene editing approach, it'd be a lot more complicated to make 37 individual drug designs, but each one would maintain the natural regulation of this gene. So kind of the implications of this, if you're doing gene addition, then you have to manufacture one drug. You are going to conduct a toxicology study on one drug. You're going to prepare and submit one IND. This is the application like to the FDA. And then if this is all successful, and you want to move this treatment from clinical trials to an approved drug, then you have a process to approve one drug to treat most or all of the patients that are affected by mutations in this gene. If you do mutation-specific treatments, then you have to manufacture a different drug, multiple drugs for each individual mutation. You would have to conduct a separate toxicology study for each one of those drugs. You likely have to prepare a separate IND and design and have a separate clinical trial for each drug targeted to each patient mutation. And then, same thing, you have a separate regulatory approval path for each of these. And so an example of, how this might apply, let's say that going back to this splicing here, let's say you have a mutation in the intron that where this intron isn't normally removed, and so you're not able to make this final spliced mRNA. This is all resulting in like a gene deficiency or protein deficiency from this gene. So you could have an option one to develop an antisense oligonucleotide or a gene editing approach to correct this very specific mutation in the intron, and you could restore normal splicing and you could treat this disease. So scientifically, like that might be feasible, but the option two would be a gene replacement strategy to treat this as a gene deficiency and add more correct gene and correct protein into the system to treat this as well as other mutations in the gene. Okay. But there are instances where, these very patient, or specific patient-specific disease mutation makes sense. And one of the areas where this has been applied a lot is in antisense oligonucleotides, or ASOs. And I think the reason why a lot of this is being done is ASO manufacturing is relatively cheap and relatively fast. I say relatively, it's because it's all relative. I'd say that in general, these ASO toxicology profiles are becoming better understood, and the design of them is getting better. And the ASO treatments can be discontinued if complications arise because you have to administer these repeatedly. And the drug is very stable, and one batch can last many years or even a lifetime. So if you're trying to develop a very individualized treatment for one patient, this is something that is happening and is kind of one of the areas where a lot of the N of one or N of few approaches are being applied. The downsides, of course, are repeated dosing is needed. Each specific mutation needs to be corrected by an ASO, by a different ASO. And each one of these would have to go through a separate toxicology study in animals every time you make a change in the design or target a different mutation. And along with this, an ASO can't always be designed. There's kind of very specific cases where the biology makes it where you could use an ASO to compensate. Okay, so now I'm going to go really into gene addition, and I'm going to use adeno-associated virus, or AAV, really as my illustration of this, where you have a viral capsid, and you've modified the original virus genome where you've taken out all the virus genes and have it package a therapeutic gene. So some practical limitations here are that the genes, I think one of the major ones is that genes are being delivered essentially as a mini chromosome. And so when the gene is in its normal location in the patient's DNA, then it's surrounded by elements that make sure that the gene is regulated and expressed properly. That all has to be kind of designed synthetically when you are delivering this in a vector. So the treated cells might express less of the gene than normal. So this could provide a benefit, but not be 100% back to normal. Or treated cells might express more of the gene than normal. And there's a possibility that this could drive kind of adverse consequences. And when we look at the vector, like the drug as itself, it's kind of the capsid is the carrier. The capsid is the delivery truck. And then the DNA inside the capsid is, in a lot of ways, the permanent drug that's maintained in the cells. So the capsid will dictate where the treatment goes and to how many cells. And then the packaged DNA is going to dictate in what cells the gene is active and how strong. So, if your capsid doesn't get to a certain cell population, then it doesn't matter what the DNA is. It's not going to express in those cells. And likewise, you might have a capsid that delivers the gene all over the place to a lot of different organs, and then the DNA design dictates that it's like only expressed in the muscle or only expressed in the liver, even though the virus capsid takes it all over the place. And so looking at the genome design, you have your favorite gene that's packaged in here, and then you've got these elements that are going to be on either side of it that are going to control where and how the gene is expressed. So it's not just that you're putting in the gene and it's just going to express exactly how it should. You actually have to design this to make sure that it expresses the right way. And then you have size limitations for AAV, and you have size limitations for just about every viral or just about every delivery vehicle. For AAV, this is a strict size limit of less than about 4,500 bases of foreign DNA. And keep in mind, that's not just the gene. You also have to account for the gene and the regulatory elements that control its expression. So if you need really complex regulation, then those regulatory elements might take up more space, and then you have less room for the gene. Okay, and what happens if you try to go beyond that? And there are approaches where to try to package larger pieces of DNA, but what basically happens is when you can design that, but then when you go to package the AAV and manufacture the AAV, then it's just going to package fragments. You might have like half of the DNA packaged in one virus and half of the DNA packaged in another virus. And those can come together in cells and recombine, but it's really, really inefficient. And on top of that, you get, the production yields go down dramatically, and the quality of the AAV preps end up going down. So this is something that can be done, but it's very inefficient, and is generally something to be avoided. And what happens if you make the DNA smaller? And there are some specific innovations that were kind of developed about 25 years ago with AAV specifically, where it normally packages a single-stranded piece of DNA, but you can trick it. You can make some changes in the AAV packaging elements to sort of trick it to packaging a double-stranded piece of DNA. And the implications of that are that when you make this double-stranded or self-complementary genome, then the AAV overall becomes much more potent, say about 10 times more potent. But the packaging capacity is cut in half. So you go from packaging about 4,500 bases to only about 2,200 bases of foreign DNA. So it also might be slightly harder to manufacture. You might get about half the production yields as opposed to a traditional AAV vector. But here I have images from a publication showing the difference of brain-directed gene therapy, where the green is showing the gene expression, and we have a traditional AAV on the left, and then the self-complementary on the right. And I think that you can appreciate that you're getting much better stable expression of the gene in many more cells. I'd estimate about 10 times more cells. And so there are a number of treatment approaches, particularly against the nervous system, where they work if you can use self-complementary AAV, but then if you move to a traditional single-stranded AAV, it's just not efficient enough. So the design of this, just an illustration of this, is work that my lab had done, led by Qinglan Ling for SIRF1 related Leigh syndrome. This is a mitochondrial disease that strongly affects the brain, but basically we made a initial design of this using a strong promoter. So we were driving strong expression of SIRF1. And this provided a benefit, but then as we dug into it more, we showed that strong amount of SIRF1 expression caused toxicity in the liver and the heart. And so it provided a benefit, but the safety wasn't good. And then we could use something that's really, really weak to drive little bits of SIRF1 expression, and that was safe, but it didn't provide enough of a benefit. And we ended up designing something that's kind of in the middle. Thinking about this as a Goldilocks of the porridge isn't too hot, porridge isn't too cold, the porridge is just right, where we had to play around with this. And so this is just a lesson learned that when you're going in and designing these, you have to put some thought behind how you're expressing the gene, and this might require a little bit of workup up front. Routes of administration, I'm just going to cover some basics here. For eye diseases, it's pretty standard now where you can directly inject the retina, or you can inject into the vitreous humor, which is sort of that jelly in the middle of the eye. There's different times when you'd want to do one versus the other, but there is a precedent of an FDA-approved drug, Luxturna, that's injected subretinally for Leber's congenital amaurosis. And I'd say that the procedure itself carries some risk. There have been some, instances of inflammation, but generally, or sort of transient retinal detachment. But generally, this has been well tolerated across trials. But there have been some exceptions. You can have direct injection into a target organ. So an example is injecting directly into the muscle, and maybe you're doing that to have the muscle become a factory to express and secrete some protein into the blood, or directly injecting into the brain. There are a number of clinical trials that have done that for things like Parkinson's disease, or now there's an FDA-approved treatment for AADC deficiency that uses that approach. And so these can be done safely, but something like a direct injection into the brain is invasive. A common route of administration for gene therapy treatments is just injecting IV, an intravenous administration, and that's obviously fairly simple. The procedure itself doesn't carry very minimal risks. And this is commonly used to deliver genes to the liver, muscle, or brain. And some examples are Zolgensma for spinal muscular atrophy, Elevidys for Duchenne muscular dystrophy, and Roctavian for hemophilia A, targeting the brain, the muscle, and the liver, respectively. One aspect of this is, especially when you're doing brain and muscle-directed therapies, these often use quite high doses. These are the highest doses of AAV that are administered across any gene therapy treatments. And as those doses have pushed higher, then this is where we've seen most of the serious adverse effects of AAV-based treatments. And so there's death in some patients due to TMA, which is basically a hyperactive immune response, and also instances of liver toxicity, which, in some patients, some clinical trials has been fatal. So I'd say, the safety issues, the majority of time, these have been well tolerated, but there are some very dramatic examples of these, very severe effects, including death. And the other thing with an IV, if you're injecting very large doses, then those high doses may be very, very expensive to manufacture, in the hundreds of thousands or even maybe millions of dollars per patient just to manufacture them. If we're moving into treating the brain, again, there are approaches to inject AAV intravenously, but what's become kind of the most common approach to treat the brain is injecting into the cerebral spinal fluid by an intrathecal or intraventricular injection. And this cerebral spinal fluid is produced in the brain, but it circulates down through the spine and back around the brain. So it kind of bathes the whole brain and spinal cord as one continuous liquid. And there is an example recently of FDA-approved drug for spinal muscular atrophy that uses this approach. And one thing that's often not recognized is, this is a treatment approach that's targeted towards the nervous system, but the nature of this is that the AAV leaks out into the blood after injecting into the spinal fluid, and pretty significant amounts of it leak out into the blood. So you still get targeting to the liver and the muscle and the heart and other peripheral organs. Doses are typically end up being lower than if you're injecting IV because the doses are really fixed around the the size of the brain. And so I'm just putting numbers here, doses as high as 1 times 10 to the 15 DNA containing AAVs per patient have been relatively well tolerated. There has been in these types of clinical trials, one patient death that I'm aware of, but this patient received a 3 times higher dose than what most of the other trials are doing. And there's also instances of possibility for liver toxicity and something called DRG toxicity, which is kind of a sensory nerve toxicity. And so really quick, if we're talking about safety, just keep in mind here, the numbers, we throw out these numbers in scientific notation, but what these amount to is, you've got, around 30 trillion cells in the body, and which is, more cells than, you would have during like a peak COVID-19 infection if you weren't immunized. That might be up to 100 billion. But a lot of these AAV-based treatments, like brain-directed AAV treatments and IV treatments, go even higher than this. If you're at 1 times 10 to the 15, that's 1 quadrillion. So it's, 100 billion times 10,000. It's an enormous load of virus particles. It's many more virus particles than you have cells in your body, just due to some of the limitations in the efficiency of AAV. And so you can do this, and in many cases it's been done safely, but patients would typically receive some type of transient immunosuppression, and also after the treatment, they would be immunized against AAV one way or the other, and that has blocked attempts so far to redose patients. So challenges of the field, I'd say that right now the gene transfer technology, there's plenty of instances where this works, but in many cases it's not adequate or it's barely adequate. And this is leading to often very high doses being used to achieve the efficacy that we want, and then that's pushing up against some safety thresholds. It also results in very high costs of manufacturing of these drugs. And again, the safety concerns, and there are still some questions about long-term durability of gene expression, like over decades. And also, not being able to redose patients is certainly a problem. So where now, is we've got the technology that we have, and I'd say most of the AAV technology, like the capsid technology, we've been working with the same technology for the last 10 or 15 years. And so we can treat patients with that technology. We know a lot about it. And there's always the question of, do you do that or do you work on better technology? And this is something a lot of patients and patient advocacy groups face is, do you kind of bet on what's working and use that, or do you go after the next best thing? And I'd say that right now we're kind of at an inflection point in the field of AAV-based gene therapy, and also a lot of nanoparticle-based gene delivery, where we're starting to have much better tools. And this is just one example here for brain-directed gene therapy, where what we've been using over the last 15 years is AAV9. And this is a new sort of modified version of AAV9, and all the purple is showing the gene expression. So this was injected at a relatively low dose where you don't see much with AAV9, but then you still get like really, really extensive gene delivery with this new variant. And so, this is a phase that a lot of patient advocacy groups and families face if they're trying to start from scratch and trying to go down a path of developing a treatment. Is, do I go with established technology, or do I go after the newer, better thing, the shiny object that's over there, on the horizon? And I'd say established technology, the first thing I just say is it good enough? If it is good enough, then you have a roadmap to follow, you have better visibility on timelines and cost. You can really plan this out and eliminate a lot of the unknowns and risks. If you opt to go towards a newer technology, it's a question, how much better are we talking about? If this is like orders of magnitude better, then maybe it's worth doing, but just keep in mind, you have greater risk. You have greater risk in terms of timelines, but greater risks in terms of costs, greater risks in terms of whether it could even be successful or ever make it into a human. Because usually we're talking about things where there's no precedent for human application. So expect that this is going to take longer, and you're going to be more expensive in the short term. But if the technology looks very promising, then it could end up being a cheaper treatment or have higher benefits in the long term. And so, long-term outlook, it might be worth investing in some of these things. And I just say, be careful. As a patient advocacy group, be careful of bankrolling somebody's research and development if the risk is high and this is very early stage, because scientists can come up with all kinds of good ideas and promising kind of early data on new technology. But if it's a long way from going to like a lab result to going into a human, and that isn't really well thought out, then you can just spend forever kind of in that R&D stage. And also I want to caution everybody about chasing headlines, and a lot of rare disease groups would be familiar with this story around baby KJ, or the first patient treated with kind of a personalized gene editing therapy. This is an amazing story, and this is really cool, and I think this is setting the stage for what future treatments can look like. But just be aware, if something like this happens once and it makes a headline, that doesn't mean that we can all of a sudden start applying it to everything. For example, like this technology would work well for the liver, but it's not ready to treat a brain disease. And it might be a heroic effort to do this as like a proof of concept, but it's not ready to roll out right now to treating thousands of different, specific patient mutations. So it's more of like a snapshot of what the future might look like, but don't just assume that there's a headline that this happened and then all of a sudden it can happen for your child. So with that, I can stop and I can take questions, and I'll put up this slide again, just that if anything that I'm talking about was confusing, then go back to some of the resources that ASGCT has that can give you a little bit more of the basics in the background. So thanks to everybody for your attention.
All right, well, now I'm glad to be joined by Dr. Gray for some live Q&A. As a reminder, you can submit questions by clicking on the Q&A polls tab, which is to the right of the video player, and then selecting Q&A. All right, Steve, first, thank you very much for your talk. That was highly informative and powerful. We do have some questions from the audience, so I'm going to get to those. So one of the themes that I've seen with the questions is around the idea of, have researchers been able to silence a mutant allele that's potentially a dominant negative or has a toxic effect, while also restoring normal function of the gene that has the mutation? And I was wondering if you could comment on that.
Yeah, no, it's a good question. And just to back up, like whenever you have one of these sort of toxic gain of function mutations, the frontline approach might be design an ASO that can target that and can just knock down and get rid of that toxic version. And sometimes that works fine. But if that gene is essential, then you knock down the bad copy, but then you're also just getting rid of all of it in general, and you could create a different type of problem. So I think that this question is really kind of addressing that latter situation, and with where this is where an ASO might fall short. And there have been examples in animal models with AAV where they can basically mimic an ASO type of approach. They can essentially express something like an ASO to knock down the gene, and then you within the same virus, you could also package an expression cassette to create more of the normal gene. So it is this sort of knockdown and replace approach. And so that has been demonstrated in animals. I'm not aware of where that's been done in humans, but conceptually it's possible as long as everything fits.
Yeah, exactly. So then another question is, if AAV delivery is not an option because the gene is too large, to fit inside of the AAV. Can you comment on some of the other technologies or up and coming technologies that could help gene delivery of these larger genes?
Yeah, I mean, there's I'd say it's kind of all of the above technologies. You know, there are other viral vectors like lentivirus that haven't, you know, haven't necessarily been applied broadly across the brain, for example. But you know, those technologies are always improving. The nanoparticle technology is always improving. I don't think there's anything like off the shelf right now, but another thing, even relying on AAV, is there are approaches to take a gene that's too big and just split it across two AAVs. And if those two AAVs go into the same cell, then you can design them so that they kind of recombine and make a, you know, a full length larger gene. Um, those approaches have really been limited because you have to have really, really high delivery efficiency to make sure that you're getting both of those into the same cell. But some of the newer AAV technology might start making those approaches possible, or making gene editing possible, you know, in organs like the brain, where, you know, I think traditionally the gene delivery has kind of fallen short of being able to get enough in there.
All right. And so we have time for maybe about one more question. Um, I guess we'll go with one that is there a definite target age to administer brain gene therapy, or is this something that is uncertain?
It's all disease specific, I think. You know, I think there's, from a gene delivery standpoint with our current tools, you know, I think there's evidence that the younger you go, then the gene delivery might be a little bit better. But most of it is really based on the disease state and trying to go in, and like most types of medicine, is better to kind of treat early and prevent things from going wrong rather than fixing them on the back end. But all of that is case by case, disease by disease.
Well said. Well, thank you so much, Steve, for sharing your deeper insights into the advancement in gene therapy approaches and considerations for patients as newer therapies arise.